Ce soir je serai la plus belle pour aller danser, danser, pour mieux évincer toutes celles que tu as aimées, aimées. Ce soir je serai la plus tendre quand tu me diras.
So now we are going to take a look at a quick survey. Actually, it's going to take a quick survey of the world of prokaryotes, in particular only focusing on our own interest in the aspect of our own interest. But anyway, these by now probably you can see, oh, this must have been the scanning electron micrograph retouched by the computerized coloration. And so these are so many different types of prokaryotes. Their 3D structures shown here. So the eukaryotes and prokaryotes, the comparison once again. So prokaryotes smaller in size and prokaryote cells do not have any internal fuzzy, sophisticated organelle structures like these, most of which we are going to take a little more detail and to learn about their respective functions is our main purpose in this chapter. But before that, let's take a look at first of this our cousin, the prokaryote cell. So overall structure, although not all the prokaryotes are shaped like this particular things, but like as a cell, prokaryote cell also must have a cell membrane. So cell membrane is also a lot of times also called the plasma membrane. So outside of this plasma membrane, unlike the animal cell, but actually the animal cell is the only exception, but all other the group of cells have additional barrier outside of the cell membrane, which is the cell wall. So cell wall is present outside of the cell membrane, the covering the cell membrane, and then serves any protection function. But the thing is that the prokaryotic cell wall is different chemically from that famous plant cell wall, and also it also different from the fungus cell wall, just like that. So the structurally they do have the same thing, but chemical compositions are all quite different is one thing. And even though they do not have any internal organelle structure, but they do have so many different ribosomes, because without them they cannot have this protein synthesis, so they cannot survive. So they must have tens of thousands of different ribosomes in there, and they don't have any nucleus, obviously. What I mean by nucleus is some structure, the membrane bound structure within which their genetic material is enclosed. That's kind of a technical definition of a nucleus. However, prokaryotes do not have them. So then how do they actually manage their genetic material, their version of a bacterial chromosome? How do they exist? Well, they have some, even though they do not have their own territory bounded by the nuclear membrane, but they do have certain their own kind of the chromosomal territory, and then we recognize that area as nucleoid. So the nucleoid is only the areas, the structure that can be found in prokaryote cell, and it is not something that defined by any boundary, but simply defined by the chromosome, that the structure tangle the structure of a chromosome itself simply just as a generated those area. In other words, just because bacterial cell doesn't have any nuclear envelope or anything, that doesn't mean that their chromosomes are all scattered over the entire the space of inside of the cell. No, they do behave themselves so that they have their own specific territory. And so outside of the cell wall, what do they have? In some cases, not all, but some bacteria have this option of having capsule, which is an additional form of a protective layer. Okay, but once again, not all bacteria have this capsule. Okay? And two different outside of this, either the cell wall or capsule in the occasion of those who have them, there are two different types of proteins, hairy protein. Both of them are kind of hair-like protein structures. One is called flagella, and the other one is called pili. Actually, the pilus is the singular, and then because it has so many, so it's a plural. Pili is used here. So flagella is also the single word. Flagella is a flagellum. Flagellum. The difference between flagellum and pili, in structurally speaking, is flagellas are longer, much longer than pili, and their functions are also different. Mostly flagellas are only exclusively used for their movement, motility, whereas pili are rather used in the attachment and some other very specific special function. So anyway, these two external structure, attachment is also kind of considered to be in the form of motility. So we can say there are two external structures for the bacterial mobility or motility function. So one is flagella and another one is a pili. Some people call this pili as a cilia because actually flagella and cilia, these two structures, can also be seen in the eukaryote too, in very similarly comparative shape. But that's something very misleading, because just because the eukaryotic cells flagellum just look like prokaryotic flagellum doesn't mean they are in same origin. Internally, structurally speaking, they are totally different type of protein. What type of proteins are actually giving the flagellum and how they are actually structured, the build is also very different. And same thing goes for the cilia. Maybe we can actually view, so then nobody actually labeled this, but maybe you could compare this in an example of such the analogous structure, maybe because of this region. But anyway, they are different. But even though the function wise in eukaryotic cell, flagellas are used for the motility, and the cilia in eukaryote, they don't call this cilia pili. Strictly they are called the cilia, are shorter, and they do have some other functions than the pure motility. Some other different special functions are actually employed for the case of eukaryotic cilia. But anyway, these two are just two different proteins present outside of the bacterial cells, and their functions are like that. And here, this photograph provides three major representative types of bacterial shape. So here one is look very ball-like shape. The other one is something a little longer, slender, plant cell-like, slender, rod-like shape. Rod. So this is rod shape, and this is the spherical shape, and this is a spiral shape. And actually, so these three are the cartonic version of three major bacterial types, the shape morphological types. One is the spherical form, we call it as a coccus, the ball-like, and the rod shape we call the bacillus. Okay, E. coli, the famous E. coli in our intestine, is the major representative of this bacillus form of. And another very long and spiral form of bacteria also do exist, and we call this a spirochete. But then out of these three basic forms, and some variant got emerged, so that a little different Coccobacillus, which is some kind of an intermediate form between Coccus and Bacillus. So Coccobacillus and Vibrio, I don't know, some variant of this Bacillus. And Spirillum is also kind of variant of a spirochete. So once again, bottom line is these three are the basic different forms of a bacteria existing on Earth. So no matter what, so imagine how many different bacteria in terms of a species must be out there. Yeah, I can countless. So many different forms of species of bacteria exist, but all of them can be either one of these three shapes. I shouldn't say one of these three shapes, because there are these three other variant forms have been also recognized. So maybe it's safer to say these 6 different forms of bacteria are basically the standard form of the bacterial shape. But still, I will stick to that. These are 3 standard. These are the 3 classical the types of standard bacterial forms. 2 different groups of prokaryotes. I've been just talking about bacteria as a single. Although, yeah, these 2 different types of prokaryotes both of them are kind of bacteria, but as we hopefully know by now, there are 2 different groups of such bacteria. One is called Archaebacteria, out of which actually we descend from. So we are the direct descendant of this Archaebacteria. What I mean by we, as we, the eukaryotes, got derived from the Archaebacteria. We got evolved from some specific form of Archaebacteria. All those evidence suggest. On the other hand, the bacteria, another group of prokaryote, we usually label them as bacteria, simply bacteria. To be most accurate, actually, they should be called eubacteria, as opposed to the Archaebacteria. But these bacteria are those that we are more familiar with because they are actually around us. In other words, these Archaebacteria are actually a little bit hard to come by. They don't really live closely together with us. If you really want to meet the Archaebacteria and then speak with them, you have to travel to some specific places. Specifically, what I mean specific, very harsh environments. So one very distinct characteristics of the Archaebacteria are their habitats are very weird. their habitats are very extreme in terms of their living condition. It's a very harsh living condition is their choice of habitat. I don't know why they actually opted to have such a very unfavorable living conditions. Maybe that might have reflect the original Earth's condition, which are very hostile, and they were the ones who initially occupied those hostile condition, and then out of them, some of the newer form of the bacteria must have evolved. I don't know actually personally, but anyway, that is the fact that we now recognize that they live in extremely unfavorable environmental condition. For example, like look at this. This is the photograph of one area of Dead Sea in the Middle East. So here the condition is extremely salty. Nobody can live. Why does it pickle the food? And if you put high salt on the food, that means in food preservation, which means if you have too much salty condition, no one can live, not even a bacteria. That's how you preserve from any bacterial contamination. But those type of method is not going to work against these guys. This is so-called halophil. Halo means salty, phil means loving. So salt-loving, halophilic archaebacteria, they have no problem of living in such extremely salty condition. Is one example of such harsh living condition. And on the other hand, like thermophil, you can imagine thermophil means high temperature, yes, heat. So they love heat. How much heat? Even almost 100 degrees Celsius, which is a near boiling point. They have no problem of living in right in the middle of such boiling water, so to speak. So in many volcanic live volcanic area or in hot spring, in this particular photograph is that of some part of area of a Yellowstone National Park, I guess. So you can see so many different bacteria, archaebacteria, around which this is hot spring, indicating that how much they don't have any trouble in living in such extremely hot temperature. On the other hand, the other extreme, cryo. We have already seen this. Cryo means some kind of a freezing. Cryophil, so the low temperature loving or freeze loving. Another name for cryophil is a cyclophil or whatever. So this pink layer are all made from the bacteria, so colonies underneath of this ice built on where I don't know where is it, Siberia or so extreme low temperature. They don't have any problem, but they can prosper under such low temperature is. And another such example, although these are not all, but only some of the chosen example of such extreme environmental condition. And this is acidophil. So like inside of our stomach, which is extremely acidic, like on pH scale of about 2, so everything will be dissolved, digested with this gastric juice, highly acidic. But then inside of such a strong acidic condition, some bacteria can live, and in the case of any different types of such an extremely acidophile archaebacteria, still they even better. They have no problem at all in living such a highly acidic conditions. So what's the takeaway for us? Takeaway of this extreme environmental condition in which they live is this for us. Who cares about whether they love such the self-torturing of anything? Good for you. But for us, it's just the enzyme, like novel enzyme, that those different type of enzyme that we never get to even imagine, or that would be possible, or such thing like that. So for example, if we had an enzyme like name DNA enzyme like DNA polymerase. DNA polymerase is an enzyme that catalyzes the DNA replication. It made so this DNA polymerase. Imagine this archaebacteria living in 100 degrees Celsius per spring. In other words, what's the reason why we cannot live in such a hot temperature? Because once we just put ourselves in, everything will be cooked. The biggest problem of this cooking is when you have this every in the morning, perhaps, like when you cook your egg fries, all those eggs get cooked up, which means all those proteins got destroyed. And in technical terms, those structural destruction, we call this denaturation. So all those enzymes will be denatured, so will be non-functioning. So then we will die, basically. Then in these bacteria living in 100 degrees Celsius, their enzymes are okay. Even their DNA polymerase will certainly survive under such 100 degree Celsius high extreme temperature. So yeah, one scientist actually they extended his idea and then actually took the DNA polymerase from such archaebacteria and brought up. And then using this, he invented this famous technique of a PCR technology. Later, maybe we should be able to look at a little more technical details of what this PCR. But we know the importance of these PCR techniques, even in this coronavirus testing and so many different areas of this PCR. Basically, what the PCR is, you hugely amplify in amount. Amplified small tiny traces amount of DNA using these DNA replication techniques, DNA replication procedure. You can amplify tremendously. That's what it is about this PCR technique. But in doing so, it involves several different transition of the temperature. You have to heat it up and then cool it down, heat it up and cool it down. And then regular enzymes from the DNA polymerase from our cells or from E. coli or anything cannot survive at each such intervals. So in other words, every interval you have to replenish or new supply of such enzyme. It will be impossible. But once you uncovered this DNA polymerase who can survive this 100 degrees Celsius, wow. It revolutionized the whole procedure. That's how this PCR technique become reality. So, and then the scientists who invented this PCR technique using this enzyme from archaebacteria first became a billionaire, and then 10 years later, eventually became a Nobel laureate. So. So many such enzymes present in archaebacteria are the major actually driving force or incentives for, of course, some other the scientists are purely curious, so academic or academically oriented and then want to learn much about the archaebacteria, but more majority of the scientists who are interested in learning this archaebacteria, because of this particular the benefits, the potential benefit of obtaining such noble enzymes and then become rich and famous. So how can they survive such extremely harsh environment condition? Is one, it's not the only reason, but mostly one major reason might be the cell membrane structure and the chemistry is very different. These archaebacteria's cell membrane chemistry and structure is different from the rest of the other organisms. So for one thing is their cell membranes, this connection and architecture is quite different from other conventional the lipid, the phospholipid, the bilayer structure of the cell membrane like this. And so the simulation study revealed that this archaebacteria, their cell membrane is cannot be really technically said of lipid bilayer, but it is a lipid monolayer, and the comparative bacteria cell membrane and phospholipid bilayer, when they were put under heat condition, soon this bacterial bilayer became just structurally very unstable and almost got destroyed. But still, this archaebacterial monolayer, without having any kind of effects, they stay very stabilized. And then a little more further heating entirely destroyed, but still the archaebacterial monolayer did not get any kind of any structural change. They are not affected. So that is a little bit of an indication, an idea, clue, giving the clue on how these archaebacteria can survive through such an extreme high temperature or lower temperature. Of course, this is not the only like a magic silver or magic bullet so that it can do it all. And having this type of a chemical structure also will have some disadvantage, like they cannot really adjust some kind of a very abrupt change in environment. So they are very poor in adapting. And actually, that's the reason why most of the archaebacteria is extremely difficult to understand and learn, because the minute they were brought up into the laboratory from their original habitat, they will die. So that has been another such a big obstacle for studying them properly. So anyway, so bacteria have cell wall, right? Cell wall is a protective barrier outside of the cell membrane, right? Of course. And this bacterial cell wall is, for example, in the case of a plant, this cellulose, which is a type of polysaccharide, which can be distinguished from very similar polysaccharide, which is a starch or glycogen, but cellulose is something different, right? But this cellulose is a plant cell wall material, but this peptidoglycan it also contains a carbohydrate, but it is entirely different from the chemical composition and the resulting structure of the cellulose-based plant cell wall. So we, without getting into any unnecessary more detail, simply put, this bacterial cell wall is entirely different from other cell walls just by looking at the name, the peptidoglycan, which kind of suggests that they are different. That's it. And also, this bacterial cell wall is also chemically very different from plant and fungi, but also archaebacteria is also have also another different in terms of a chemical structure, different from that of the bacterial cell wall. So they are all different. So do all bacteria have the cell wall? There's one exception. The Mycoplasma is a name of bacteria. It actually probably represents smallest bacteria other than the artificial bacterial cell made by that Craig Venter that I mentioned in the previous chapter. Right? So this Mycoplasma somehow does not have any cell wall. So, and because it doesn't have any cell wall, that famous antibiotics penicillin. Why is penicillin famous? Because it was the first ever produced antibiotics, and that one that saved huge, tremendous lives ever since its production. The penicillin has been the lifesaver. Before the era of penicillin, a lot of people have to die once they get infected from bacteria, right? So when you have this bacterial infection, there's no other way but your immune system can somehow recover. So that's why actually 1/3 of, for example, entire population, European population, got wiped out by Black Death and in the earlier days, and also in those especially in World War One, a lot of those the soldiers got wounded out of the battlefield. Even if it is a relatively minor injury, they have to get through this amputation because of the infection. So this antibiotics has been really such a great invention, probably after the invention of the wheel. But this Mycoplasma is not like penicillin is not effective against Mycoplasma because it doesn't have any cell wall, which indicates what? This penicillin is targeting the bacterial cell wall. So this Mycoplasma doesn't have any cell wall, so there's nothing. For the sake of Mycoplasma, if you were a Mycoplasma, why do I have to fear for penicillin? Because nothing can harm. I have nothing that can be harmed by penicillin is the idea. And this is the figure. Here, this big ball is a red blood cell, and the small purple dots are each individual Mycoplasma bacteria that are trying to infect our precious red blood cells, the photograph. Okay, so that's about the cell wall. And what about the capsule? I said this capsule isn't an optional thing. So not all, but some bacteria have an additional protective layer called the capsule. And this capsule, on the other hand, is something different chemically from the cell wall, and it is basically mainly made of carbohydrate, sugar. It's a kind of a sugar coat. And so here, capsule, outside of cell wall, there is a capsule like that. And what good would you have if you have the capsule additionally? Well, as a bacteria, if you have a capsule, it's really such a nice option, because once you are protected by capsule, then you will become more virulent. Virulent means more pathogenic, sort of saying more deadly. It's much more difficult for us to eliminate once they got inside of us. This means virulent. Why? Because The presence of capsule make this bacterial body more slippery. Like, if not, yeah, whether you have a capsule or not, if we have an antibiotics drug, then we will be able to take care of those bacteria. But let's say without any the help of the antibiotics, we try to fight off on our own immune system, then this capsule bearing bacteria become more problematic, in that the way we usually take care of this bacteria is our white blood cell one specific kind of many different white blood cell. We do have the cell eating cell. One of them is called the macrophage. This white blood cell can capture any invader and then engulf, eat it up, and then inside it digests all those the invading cell. That's how we eliminate any foreign invader. And then this try to capture this hands, extending hands that's being projected from this macrophage, the white blood cell. Then this presence of the capsule make this capturing step very vulnerable to like have this slip away from this grip because of this slippery carbohydrate surface. That's how they actually more easily evade from this the defense action. And speaking of this, let's go further and then talk more about the cell wall, bacterial cell wall. There are two different types of bacterial cell walls. So before we were able to classify all those living bacteria according to the shape, outside of shape morphology, into standard 3 different, or like if we consider some variant, up to mostly like 6 different the classes. Here, by the specific type of a cell wall architecture, we can divide the entire all living bacteria into simply two groups. One is gram, so-called gram positive. The other one is gram negative. By the way, this gram is the name of the renowned microbiologist who invented this specific way of staining. So gram is a staining. That was invented by this scientist Gram, his last name. So simply gram positive means those bacteria who respond favorably to this gram staining method, and gram negative bacteria are all those the other type of bacteria who is very resistant to the gram staining. Why is that so is actually the reason why we are looking into this. The structural bacterial cell wall structures are in these two different alternatives. In the case of a gram positive, they do have this. Look at this gram, the cell wall. This peptidoglycan cell wall layer is huge. I mean thick, very thick peptidoglycan cell wall layer. In other words, their protective layer is really reinforced, thick, hard protective layer. So they are basically safe. And this peptidoglycan, this chemically speaking, if you think about it, this peptide, amino acid, and glycan is carbohydrate. All of them are, do you think these materials are hydrophilic or hydrophobic? Yeah, I would guess it's hydrophilic. Yes, it's polar. So this staining material will get easily be attached to this peptidoglycan layer. So that's why this gram-positive bacteria is so positive to the gram staining. So that's why they can be labeled as gram-positive. So the only actual real reason why this gram-positive bacteria is a gram-positive is because of this thick, very thick, their peptidoglycan cell layer. But on the other hand, another group of bacteria, regardless of the different type of species, they do have this peptidoglycan layer is very thin. Oh, look at this. Compare this with the gram-positive. How can they manage? How can they function with such a thin slice of the protective barrier, cell wall? Aren't they, supposed to? Aren't their cell wall layers supposed to protect their whole entire surface? And do you really think that such thin layer of a cell wall would be enough to provide this proper protection? Yeah, probably those gram-negative bacteria also would have agreed. Okay, this is something like untrustworthy. I cannot really put all my eggs on your basket. So they have they employed additional cell membrane, outer membrane. So instead of having this such a thin cell layer, they have one extra plasma membrane layer outside. So they do have this so-called outer membrane and inner membrane. These two different types of cell membrane layers they have. So the cell membrane, as a cell membrane, it's a lipid bilayer, phospholipid bilayer. So it is basically hydrophobic. That hydrophilic staining solution will not be able to actually stick easily together with them. That's one major reason why this gram-negative bacteria is a gram-negative. And another thing is, even if those stain solution was able to succeeded in penetrating the first cell membrane barrier, this real region of the peptidoglycan layers are so thin, or maybe should I say too thin, so they won't have any too strong or an impressive enough staining remain over here. That's why they are gram-negative. So once again, so gram-negative bacteria have one additional outer cell membrane. Why? Because the peptidoglycan cell wall is too thin. So this outer cell membrane is a kind of a necessity to become adequately the protective. And in addition to this outer cell membrane, they do have this LPS. This is something that present on their additional outer cell membrane. This LPS layer is something that unheard or unseen in the other type of bacteria, which is gram-positive. So this LPS stands for lipopolysaccharide. So probably you could guess what type of material is this. Lipid, lipo, and the polysaccharide. So this is basically containing some polysaccharide plus lipid. And the reason that I have just explained, so this gram-negative bacteria is not really stained very good with this gram staining because of this lipid containing layer. And this LPS, the lipopolysaccharide layer, is also known as something in endotoxin, or more professionally is called pyrogen. So this endotoxin or pyrogen is something very serious problem in those who are working in the animal cell culture as well as in the medical field, is because what does it do is, in this endotoxin, is Why is it called an endotoxin? Is because probably it is some kind of an internal part of this bacterial cell, and it is acting as a toxin. That's why it is an endo, inside, means endotoxin. But more importantly, why this endotoxin, which is like basically it was originally derived from the bacteria, gram-negative bacteria LPS, is because this LPS is tightly linked with the inflammation. This inflammatory response is this. And we now know how deadly this inflammatory response can be. We learned from the case of this coronavirus infection. Actually, majority of those patients who died is not because of the actual direct toxic effect of a virus themselves, but our secondary response involving this. The inflammatory response is the main cause of this death, and all other, like the pain and the troubles that we have to go through once we get infected from the coronavirus. So this inflammatory response is basically what it is our overall reactive immune response mediated by a whole bunch of the white blood cells, and then it specifically result in some tissue damage in specific area. But this LPS is triggering this. So here, in the one such an example of this, our white blood cell driven inflammation response, which is originally caused by this LPS, which initially bind with some of the receptor. It's always kind of receptor and the receptor which is present on our cell surface, and the binding molecule specifically that interact with the specific receptor is called, its name is ligand. So the LPS is acting as a type of ligand for, in this case, of the immune response. And that's how you escalate this deadly inflammation response. And then some of our endothelial cells got damaged. And similarly, our brain cell also can be damaged by this LPS taking these three specific steps of the procedure involving the white blood cells, although we don't really have to go into too much detail. So the bottom line is LPS, which is present on the surface of this gram-negative bacteria, is acting as like a turn-on signal for very specific immune response, which will result in the inflammation response, which will damage our many areas of the tissue. That's why this LPS is considered to be a toxin, and to be more specifically, is an endotoxin. Okay. Now, gram-positive bacteria, it distinguishes from the gram-negative bacteria by having very thick cell wall. So because of that, they don't have to have any additional outer cell membrane. So they do have any LPS. That's simple as that. And because of that, speaking of this gram staining, it has very specific conspicuous staining in the gram staining reaction. So these are once again like a reiteration of the difference between the gram negative having LPS and gram positive, and an actual staining result can you distinguish is at best Like Pseudomonas and E. coli are the two examples of such gram-negative bacteria. Although I really hope that you don't really even think about it, like memorizing. Oh, Pseudomonas and E. coli are gram-negative. No, that's useless. Please don't do that. And on the other hand, Bacillus and Staphylococcus are the other good example of such a gram-positive bacteria. But then these are the one examples, Larry, a difference between these in gram staining. Okay, so that's about bacterial cell wall. Now another interesting feature that only bacteria have is also Archaebacteria also have too, and prokaryote have is something called the bacterial plasmid. Okay, so what's plasmid? Plasmid is a DNA, but the DNA is something extra chromosomal, something like a satellite. Something like satellite DNA is an extra chromosomal DNA, which means your DNA is always like organized into the structure we recognize as a chromosome, but outside of this chromosome you have also some kind of extra DNA, and it can be found in almost every bacteria, and they are recognized as a bacterial plasmid. Although some yeast is an eukaryote, but some yeast also have some plasmid, but not as much extensively as the case of the bacteria. So we just like take that example as an exception. So bacterial plasmid is something that uniquely found in Eubacteria and Archaea only. So like if in this cartoon, if you consider this as their main chromosome, but outside of this, much smaller, tinier actually, in more realistically, the size comparison is much more greater, and this is an kind of a circular. Once again, this is circular form of the DNA, which is a double helical form of DNA. So all this, whether it is a bacterial chromosome or bacterial plasmid, they are all circular. It is a kind of a characteristic, unmistakable characteristic of prokaryotic DNA. Prokaryotic DNA. They are all circular, whereas eukaryotic DNA is a linear. And because of this linear configuration, we have our own kind of a destiny, fate, or some serious fate. Later, hopefully, we can touch this problem. So this plasmid is once again, if we kind of correctly define, it can be said this small, circular, and autonomously replicating DNA. So in other words, this plasmid does not have to get any permission before it replicates its own DNA, permission of the main guy, the main chromosome. Hey, when are you going to replicate? So can we replicate together when you are about to replicate? No, it does not have to. Actually, in the case of a chromosome DNA of a bacteria, like how many times do you think in its entire lifespan of a bacteria, how many times do you think the bacteria chromosome can replicate? Only once, just right before the cell division. Once the one bacteria gets split into two bacteria, then its previous lifespan is over, right? So during the single lifespan of any individual bacteria, a chromosomal DNA will replicate only once. However, in the case of this plasmid DNA, no, it does not apply to this once in a lifetime rule. Usually the plasmid DNA replicates several times, so many times within a single lifespan of a bacteria. Okay, that is something that we recognize as it replicates autonomously. Okay, and it is extremely small, comparatively speaking, with the bacterial chromosome and DNA. And another thing is a copy number. Bacterial chromosome exists only in a single copy in a cell. But the plasmid, in the case of plasmid, it can exist. Usually, naturally, it exists in the several, like tens of 20s or several hundred. But human, always this human intervention and manipulation created some kind of monster for our own, to use in our own advantage. So we have created some kind of a version of a bacteria and a kind of a version of a plasmid that can exist in something like up to 5,000 copies in a cell. Wow, it's a huge, isn't it? In a single cell, this exactly identical copy of a single plasmid that can exist in 5,000. Although it is not natural though. So the multiple copy is another key. It's something kind of a little bit reminiscent of the mitochondrial although this mitochondrial DNA is a prokaryotic DNA in circular DNA, but the size is usually rather much larger than usual plasmid DNA. And although the mitochondrial DNA can also exist in multiple copies, but the copy numbers are no comparison. This plasmid, in the case of plasmid, usually much more number of copies exist present. So like this figure, what this figure is trying to convey, the message is here there is a bacterial DNA in the bacterial chromosome, although even if it is a circular still, because it's too large, it's several bends and turns of this shape. But in the case of a plasmid, here it's trying to at least portray two different types, two different kinds of plasmid in terms of this different size, trying to indicate that the single bacteria does not really have to carry only one kind of a plasmid. It can carry several different kinds of a plasmid within a cell, each of which can be present in huge multiple copies is the key. And when they replicate, of course, when the bacterial DNA, single bacterial cell split into two, it goes through their own DNA replication so that they can be partitioned into two new cells equally well. Why are we looking at this plasmid? Because there is a reason. Let's just take a look at the good side first. So this bacterial plasmid has been really useful, instrumental in the progress of so-called genetic engineering technique. The genetic engineering technique, another terms describing such genetic engineering technique is recombinant DNA technology, the same thing. So this figure is basically trying to illustrate the process, nuts and shells of the recombinant DNA technology. And this is actually a real example of what initially just has happened in something like in 1975 for the first time in the whole new world. That was the first instance of such genetic engineering occurred. So using the plasmid here, some scientists tried to had an idea of specific gene of interest from the human chromosome. Actually, that chromosome, the gene, happens to be on a insulin gene, human. Before this attempt, like the only source of having this insulin, obviously we cannot extract the insulin from the human, so we were obtaining the insulin to cure the diabetic patient from pigs and some other related animals. Probably that's pigs were the only animals that provided the source of insulin before this type of techniques actually ever attempted. Then he thought about taking this human insulin gene, so you have to precisely excise, cut it out, the insulin gene. To do so, actually, you needed some scissor, like a molecular scissor. Of course, you need the molecular scissors to precisely excise the boundary of this insulin gene. And fortunately, we also had that those enzymes available, which were also stolen from bacteria. We will talk that later. And so took this insulin gene out and then inserted it over here. So now this plasmid became a chimera. This plasmid originally was actually all bacterial DNA, 100%, but then it's got the portion of this human genes got inserted over here. So that's why I call this now is a chimera. And that's why it is called some recombinant DNA, right? Like reshuffled, the re-engineered DNA. And now this plasmid, we can tell plasmid to go back to your home, original home, which is inside of the bacteria. Now inside of the bacteria, this plasmid will replicate as normal as before, and better yet, this gene, human insulin gene, although it isn't human insulin gene, which is a part of eukaryotic gene, can be properly recognized by bacteria and then processed, so that insulin protein is produced by the action of this DNA, chimeric DNA, recombinant DNA. So that's the first incident of the genetic engineering. So then, using this, basically this technology these days, that's how we produce the insulin so cheap. So that's one such an example of recombinant DNA. So in recombinant DNA, this plasmid DNA serving as the vector for accepting the foreign DNA and then storing them, and then they can be put into the host, the expression host, and then do the respective purposes. Well, I have to stop here. Although actually in original recording I went through ahead and all the way up to like almost finishing the antibiotics resistance, but still were not able to finish what I had intended, like at the end of this prokaryotic discussion. So then I kind of anticipated that it's not going to I'm not going to be able to finish all those things even within an hour and 40 minutes. So that's not going to work. So I decided to go back and then stop at this point, and then I will pick up the rest of this after the midterm exam, and then we'll just combine them with the new eukaryotic discussion of the chapter. So that's probably I will have to. So exam will cover up until now, and I will shut up here, and hopefully you will have every one of you have the best of your luck in the upcoming exam. So